Dear students, in this module, I'm going to talk about improving the performance of the Chow-Fasman algorithm. The Chow-Fasman algorithm, in its classical description, predicts alpha helices, beta sheets, and beta turns from the sequences. So how can we improve this algorithm further? For that, we need to first remember that this algorithm is based on the statistical occurrences of various amino acids within the different secondary structures. So therefore, to improve this algorithm, to improve the predicting power or the performance of this algorithm, you need to improve the foundation of this algorithm, that is the statistical numbers that you obtain from the PDB. So towards that, the secondary structure propensity values for each one of these secondary structures can be recalculated. Remember that the Chow-Fasman algorithm was developed several decades ago, and now we have many more structures that are available within the PDB. So if you take all of those newly reported structures and you see which amino acid occurs in which secondary structure within the overall structure of proteins, then you can have an improved set of statistics that can help improve the performance of the Chow-Fasman algorithm. Next, you need to consider that not all amino acids and the propensity for their occurrence within the secondary structure specifically defines their existence. The statistics or the propensity values are just an average for each amino acid's occurrence in various secondary structures. So if you want to specifically find out which amino acid occurs in which secondary structure in a specific type of protein, you may want to look at several of its properties, such as the nucleation region within the proteins, the hydrophobic cores. So for example, if an amino acid occurs within the hydrophobic core, then you may want to incorporate that information into the secondary structure prediction as well. Of course, the membrane proteins are also unique, and the propensities that we calculate from the PDB are not reflective of the specific properties of the membrane proteins. So you may want to include such information into the predicting algorithm as well. Next is the point that for the beta turns and their prediction, we only consider 4 neighbors from a residue in the Chow-Fasman algorithm, that is Fj plus 1, Fj plus 2, Fj plus 3 besides Fj. So using these tetrapeptides, we compute the propensity for forming a beta turn. But as you know, the loops can have a variable size. So if the loops have variable sizes, then we cannot rely on just 4 amino acids, and it may be useful to consider variable size loops to form these structures. Next is the conditions that are surrounding the protein while it is folding. So not every amino acid behaves in exactly the same way in all environments. So depending upon the environment, a polar amino acid, for instance, may behave differently. Therefore, you need to consider which environment is provided to the protein for folding. Moreover, the solvent accessibility to the residues may also be considered towards improving this algorithm. If you know which organism the protein is from, then you can also see how the structure of that protein is different for that specific organism. In the classical Chow-Fasman algorithm, that is not considered, and proteins from different organisms are averaged together towards calculating the propensity for each amino acid. Therefore, towards improving the performance of this algorithm, you may want to look at the specific organisms from which certain proteins come. So in conclusion, the Chow-Fasman algorithm, despite the fact that it works very nicely, it can be improved further by considering the hydrophobic properties, the location of proteins, the accessibility of the solvents, the organisms, and so on and so forth. By incorporating this information, you can better predict the secondary structures that can be formed out of the primary amino acid sequences.